Today we will start with the Alkyligen faecalis or Alkyligenaceae family, which is the most important organism in Alkyligenaceae is Alkyligen faecalis. Alkyligen faecalis, there is another species that is called Acromobacter, and Acromobacter is also referred as Alkyligen pichendi. So this group is not confirmed in microbiology identification chart. This Alkyligenaceae family contain the most important bacteria, that is called Bordetella. That Bordetella is the major cause of whooping cough in children, and the organism is non-motile, weakly catalase and oxidase positive. There is Bordetella pertussis is the major organism found in the children, which is the nasopharyngeal specimen, and aspirate from the young children are preferred, which is the you can say the specimen of choice. The organism is oxidase positive, but do not grow on SBA. They have a pertussis toxin. We can also do the as a gram-negative bacilli, or we can directly do the direct fluorescent antigen test. The organism is very fastidious, so they need a transport medium that is called casemino acid broth. The casemino acid broth contain charcoal to absorb the toxic substances associated with the swab. The specialized medium for the Bordetella pertussis is BGA, that is Borget gangrel agar potato infusion. Is made of potato infusion, and they have a murky drop like colony spelomorphic in shape. If we see here, we describe that it is a gram-negative cocobacilli colony, entire dome shape, gray, shining, resembling drop of mercury. The another species is a parapertussis. Parapertussis grow on SBO, but it will take 2 to 3 days, and oxidase negative, and urea is hydrolyzed within 24 hours. The animal pathogen in this family is Bordetella bronchoseptica, which is an animal pathogen mostly causing cough in the dogs, and oxidase positive and grow on SBA within 24 hours, and the rapid hydrolysis of urea within 4 hours. And another species is Tremetum, Bordetella tremetum, causing the ear and wound infection. The cephalosporin third generation ciprofloxacin and erythromycin is the drug of choice for this organism, inhibiting the epithelial cell in the upper respiratory tract of the man, to which the bacteria is attached, invade and survive, highly contagious, transmitted through respiratory droplet, apple green fluorescing, using the direct antibody fluorescent test, which is a rapid test to detect for the detection of a Bordetella pertussis infection. Then we will go for the Acromobacter. Acromobacter, as I explained, there is this is recently classified into Acromobacter. Before it is been classified with the Alkyligen family, like Alkyligen faecalis, Alkyligen pichendi, Alkyligen dentrificans. Now it is Alkyligen Acromobacter dentrificans and Acromobacter pichendi, and this nosocomial pathogen contaminate the solutions and mostly found in the cystic fibrosis patient. It's a motile, catalase, oxidase positive, grow McConkey, SBA in chocolate, white to 10 colonies within 24 hours. Same like Alkyligen faecalis, they can also motile, catalase, oxidase positive, gram-negative bacilli, but they are little large in size and grow well on McConkey chocolate and SBA within 24 hours. The main identification marker for Alkyligen faecalis is that the organism have ability to directly convert nitrate to nitrite gas, and motile by peritracheous flagella and apple odor and green discoloration, acyclolytic and found in the blood, sputum, urine, and nosocomial infection like nebulizer, respirator, lavage fluid. Drug of choice is piperacillin, tazobactam, and clavonic acid, because the organism is also able to produce enzyme that is called penicillinase. Another group of the bacteria which is very important, that is a normal flora of oral cavity, and that is called Hack group. Hack group is a fastidious organism group, and that need 2 to 4 days at 35 to 37 degrees centigrade with 5 to 10% carbon dioxide or microaerophilic condition. Candle jar, which is very important because the candle jar provide the moisture. if we see in this organism, in this chart, we classify organism on the basis of their biochemical test, which the first one is oxidase and nitrate. So when the oxidase is positive and nitrate is positive, the first organism in this group is Actinobacillus, which belong to the Pasteurellacea family, and that is usually this organism is usually transferred by animal bite or a contact and cause the disease in immunocompromised patient. Usually it is a gram-negative Coccobacilli, can grow on chocolate and SBA, pinpointed colony, non-hemolytic. And along with the oxidase and nitrate, the another biochemical test, which is urease, that is also positive for this whole group. In this whole group, 3 organisms are there. One is Actinobacillus equilis, which is along with the oxidase, nitrate and urease, H2S, and ONPG also positive, and SUS is acetylcholine positive and ONPG positive. Whether if we see the Actinobacillus uri, that can grow on the McConkey, which differentiate this organism from other species. Another organism that is oxidase and nitrate positive, that is Echinella cordon, that belong to Neisseria family, found in oral cavity and dental plug and sublingual respiratory secretion, wound and blood infection. And basically the organism is a normal flora of human, and it is associated with the trauma due to the human bite. 2 to 4 days under 5% carbon dioxide at 35 degrees centigrade. Organism required the hemine factor, X factor, and also produce the pit and bleach like odor in surface of a agar, chocolate agar. The organism is nitrate and oxidase positive, but negative to catalase. This is the drug of choice for this organism is penicillin.